Welcome to the Explainer. Today, we are going to completely change how you look at a simple glass of milk, or, that pat of butter on your toast. We're diving deep into the architecture of milk lipids. Now, most of us just think of milk fat as a dietary thing, right? It's the stuff that makes ice cream creamy or just adds calories to our day. But if we look at it from a biological standpoint, milk fat is this highly engineered, dynamic biological blueprint, and it's perfectly tailored for the survival of the next generation. So let's jump right into this hidden biochemistry and see exactly how nature constructs these really remarkable molecules.
To kick things off, I want you to just think about this for a second. Why does a whale produce milk that is over 50% fat, while human milk hovers right around 4%? It's a massive contrast, but it beautifully highlights the core biological purpose of lipids. I mean, all mammals produce milk with fat in it, sure, but the concentration is entirely dictated by the environment. For marine mammals like whales and seals, their newborns need immediate intense energy just for thermoregulation, just to survive in freezing waters. Since fat is literally nature's most compact energy storage, their milk is basically engineered to be a biological survival suit. So, here is our game plan for today. First up, we'll look at the blueprint of milk fat. Second, the triglyceride molecular scaffold. Third, the minor lipid players. And finally, we'll wrap up with the biological fat factory.
All right, part one, the blueprint of milk fat, how different species actually tailor their dairy. The sheer scale of variance across species is just fascinating. You've got cow milk sitting between 33 and 47 grams per liter. Human milk is pretty much in that same neighborhood at 38 grams per liter. But then check out the harp seal. Its milk is packing an astonishing 502 to 532 grams of fat per liter. That's over 10 times the concentration. Again, this is thermoregulation in action. The blueprint of harp seal milk prioritizes this massive fat transfer just to insulate the pup against the icy Arctic environment.
And what's really cool is that even within a single species, like the dairy cow, this lipid blueprint isn't just some static thing. It's incredibly dynamic. Looking at the macro to micro level, we can see a cow's fat content is influenced by a ton of factors. Breed plays a massive role, for sure. A Jersey cow naturally produces a higher fat milk, around 4.5 to 5.5%, while a Holstein is closer to 3 or 3.8%. But it's also heavily influenced by individual genetics, exactly what the cow is eating, and even the stage of lactation. Like the hind milk at the very end of a milking session actually contains way more fat than the foremilk at the beginning. It's a constantly adjusting living system. Moving on to part two, the triglyceride molecular scaffold. These are your core structural building blocks. Okay, so when we talk about milk fat, an overwhelming 97 to 98% of it is made up of triglycerides. Now, I want you to think of a triglyceride's biological scaffold kind of like a molecular coat rack. The vertical post of the rack is a molecule of glycerol, and it has 3 very specific hooks where it holds on to 3 fatty acids. We call these ordered positions SN1, SN2, and SN3. And here's the thing, nature doesn't just throw fatty acids onto this rack randomly. Not at all. For example, stearic acid is almost always found at the SN1 position, while palmitic acid favors that middle SN2 spot. And those short-chain fatty acids, they almost exclusively hook onto the SN3 position at the bottom. So you're probably wondering, why does this exact molecular architecture actually matter in the real world? Well, first off, this non-random, super-organized placement completely dictates the hardness and the melting point of your dairy products. If you were to somehow chemically randomize these positions, the physical hardness of your butter would totally change. Secondly, it's absolutely critical for digestion. When a newborn, or even when you drink milk, digests this fat, an enzyme called pancreatic lipase acts like a pair of biological scissors. But it specifically targets the SN1 and SN3 positions first, because nature cleverly placed those water-soluble, short-chain fatty acids right at SN1 and SN3, they get snipped off immediately, rapidly absorbing to give quick, life-sustaining energy to the baby. It is just an incredibly elegant design, don't you think? All right, let's look at part 3, the minor lipid players. These are the critical 1% finishing touches. Now, triglycerides might hog the spotlight at 98%, but less than 1% are phospholipids, alongside a tiny fraction of a percent of cholesterol, vitamins, and other trace lipids. But whatever you do not let their small numbers fool you. These minor lipids punch way above their weight class when it comes to dictating the structure, the functional properties, and the health benefits of milk. Chief among these minor players are the phospholipids. They form something called the milk fat globule membrane, or MFGM. See, inside the milk, the fat doesn't just float around unprotected. It's entirely encased in this delicate phospholipid-rich protective bubble. This membrane is what stabilizes the fat emulsion. It keeps the fat uniformly mixed in the milk instead of just instantly pulling at the top. And here is a super fascinating processing fact for you. When you churn cream to make butter, that physical agitation actually shatters this membrane. The fat clumps together into butter, and those highly functional membrane fragments, which are super rich in phospholipids like sphingomyelin, get washed away into the leftover liquid, which, by the way, is exactly why traditional buttermilk is so incredibly rich in phospholipids.
Beyond phospholipids, that remaining fraction includes some pre-recognizable names. Cholesterol is in there, but honestly, it sits at a very low level, only about 0.3% of the total fat, basically just acting as a structural component for the membranes. Then we have carotenoids. These are the hydrocarbons that literally give butter and milk that signature yellow hue, while also acting as a crucial precursor to vitamin A. And rounding out the trace players, we've got our fat-soluble vitamins, A, D, E, and K. And the levels of these are highly variable depending on exactly what the cow is eating and her stage of lactation.
Which brings us to our final section, Part 4, the biological fat factory, how we assemble milk fat right from the blood.
First, we have to look at the raw materials, and honestly, this differs drastically depending on the species. For non-ruminants, like us humans or pigs, our biological factories primarily run on glucose to synthesize fatty acids. But ruminants, cows, goats, buffalo, they have a completely different engine. Their primary precursors are acetate and beta-hydroxybutyrate. These compounds are produced in the animal's rumen, which is their specialized stomach, by microbes that are actively fermenting the rough, fibrous plant material the cow eats. This is literally why a cow fed a low-fiber diet will actually produce milk with a lower fat content. They just lose the microbial acetate they need to fuel their fat factory.
And this reveals perhaps the most mind-blowing fact of this entire explainer. Where does the total fat in a glass of cow's milk actually come from? Well, it's roughly a 50-50 split. About half of the fatty acids, specifically the short and medium chain ones, are built entirely from scratch right there in the udder's cells. Scientists call this de novo synthesis. But the other half, the longer chain fatty acids like stearic and oleic acid, those are harvested directly from the cow's circulating bloodstream, originating straight from her diet or her body reserves. So how exactly does the udder harvest those circulating fats from the blood? Enter lipoprotein lipase, or LPL. Think of LPL as a pair of biological scissors stationed right on the walls of the mammary capillaries. After a cow digests her food, dietary fats travel through the bloodstream packaged up in little vehicles called chylomicrons. As these chylomicrons float past the udder, the LPL scissors activate. And actually, their activity increases 8-fold during lactation, which is wild. And they just snip the long-chain fatty acids right out of the passing bloodstream. Then they get absorbed into the mammary cell and added straight onto our triglyceride coat rack.
And this brilliantly illustrates the entire chain of causality. It connects the macro world of the farm directly to the micro world of biochemistry. Step 1, the input. The cow eats a high roughage pasture diet. Step 2, the engine. Microbes in the rumen ferment that fiber into acetate. Step 3, transport. Precursors travel through the blood while our LPL scissors harvest the long-chain dietary fats. Step 4, the factory. The mammary cells take those harvested blood fats, synthesize the rest from scratch using acetate, and arrange them perfectly onto the glycerol scaffold. And step 5, the output. We get a highly specific triglyceride profile that ultimately dictates the exact hardness, flavor, and melting point of the cheese or butter sitting right there on your table.
So as we wrap up this explainer, I want to leave you with this final, kind of provocative thought. How does a cow's daily fiber intake ultimately re-engineer the microscopic architecture of your very next stick of butter? It's all connected. The farm environment directly programs the cellular factory, determining whether the SN1, SN2, and SN3 positions get the exact fats they need to create a perfectly structured lipid. It just proves that food is never just an ingredient. It is a continuously evolving, responsive biological system. Thanks so much for joining me today, and I really hope this deep dive leaves you curious to explore even more about the incredible science hidden inside our everyday food.